Hi, today we'll talk about winemaking, but not really about the technology of winemaking or how to choose a good bottle of wine, something that I know little about. Instead, we talk about the genetic basis of winemaking. Humans figured out how to make wine thousands of years ago, but with all due respect, the biggest star of winemaking is not Robert Mondavi. The biggest star of winemaking is yeast. However, until recently, it was not clear how yeast accomplished the very complex process of winemaking. And today, we will learn how clustering algorithms help scientists to figure out how yeast make wine. I'll start with the question which domesticated animal is missing at this slide. And of course, you already know the answer since the whole lecture is about yeast. It is yeast. In fact, 10 years ago, scientists found an Armenian cave equipped with all winemaking equipment, including even stone cups for wine drinking. And my next question is, why do we store grapes in sealed containers to make wine? It doesn't appear to be necessary because yeast live on grapes and they have natural ability to transform glucose in grapes into ethanol. And therefore, it looks like the only thing that is needed to make wine is simply to crush grapes and let yeast do their job. However, it's not that easy. It turned out that yeast have developed another amazing ability. After they transform glucose in ethanol and run out of glucose, they can invert their metabolism and feed on the ethanol they just produce. And thus, they would ruin the wine that they just made. And this change, reversion of metabolism is yeast is called the diauxic shift. And our goal today is to figure out which yeast genes are implicated in the diauxic shift. But before we answer this question, I wanted to ask you, what does it take biochemically to convert glucose into ethanol? Imagine the time over 100 million years ago when the first fruit-bearing plants appeared on Earth. At that time, an organism that it would invent a way to metabolize glucose in fruit would have an enormous evolutionary advantage, particularly if it would convert glucose into ethanol, because ethanol is a poison for most other competitors of this organism. However, transforming glucose into ethanol is not a small feat, let alone transforming ethanol, metabolizing ethanol afterwards. It is a very complex biochemical reaction, and it requires creating new metabolic pathway, essentially a revolution in biochemical abilities of the organism. We know that genes mutate and can build new function, but in this case, developing 1, 2, 3, even 10 genes here and there won't accomplish the task of building a new metabolic pathway. That creates a certain evolutionary puzzle, and we'll return back to Susumi Ono, whose work we studied in genome rearrangement chapter. Susumi Ono came up with this random breakage model that has become the dogma of chromosome evolution study for 30 years until it was refuted just 10 years ago. So why do we bring Susumi Ono if his random breakage model turned out to be wrong? Because he came up with another evolutionary model that had a very different fate. He argued that big leaps in evolution would not have been possible without whole genome duplication, essentially doubling the number of chromosomes in the genome and thus doubling the number of genes. And whole genome duplication, in difference to random breakage model, first was heavily criticized by biologists. They did not believe into it for 30 years until It was confirmed just 10 years ago, roughly, at the same time when the random breakage model was confirmed. But how whole genome duplication helps is to invent new metabolic pathway. Well, right after the whole genome duplication, every gene will be present in 2 copies. One copy will be free to evolve whatever new revolutionary function is needed, for example, creating a new metabolic pathway, while another copy of the gene will simply support the life of existing algorithms, do whatever function it was supposed to do before the duplication. So after we introduce this biological background for the diauxic shift, our next goal is to explain how biologists measure gene expression to study the diauxic shift.